My name is Josh Bradshaw, and I worked in the Giovannoni lab this summer, and my mentor was Julia Vrebelov. And in the Giovannoni lab, we study ripening in fleshy fruits using the tomato as a model system because it grows quickly in the span of 3 to 4 months, and its genome is very well characterized, which allows us to transform certain genes. And one mutation that I focused on in ripening this summer is called the RIN mutation, the ripening inhibitor. And in this fruit, in this phenotype, all ripening is completely inhibited, including ethylene production, volatiles, softening, and carotenoid biosynthesis. So the actual RIN mutation is a deletion in the RIN and microcalyx region of the genome. The RIN gene is a transcription factor which regulates genes related to ripening and allows the tomato to become appetizing and appealing for animals to spread its seeds and allow the plant to reproduce. So in the RIN mutant, there's a deletion in the 8th exon of the RIN gene, as well as the first exon in the microcalyx gene, which is the gene adjacent to it on the chromosome. And it results in a fused chimeric message of the two genes. And the protein that this gene makes does not work to turn on other ripening genes, which leads to a severe phenotype of an unripening tomato that will remain green for 8 days, 15 days, 60 days. It remains green terminally and unripe. So it was suggested that this severe phenotype was due to a repressor in the 8th exon of the microcalyx gene called the EAR domain. So CRISPR lines were generated, which would essentially knock out the EAR domain, as well as another CRISPR line was generated to knock out the 3rd exon of the microcalyx gene and erase everything downstream of it on the chimeric message. And so the CRISPR lines are shown here. This is the line that targets the EAR domain. These two basically target the entire 8th exon of the microcalyx gene. And this CRISPR line just targets the 3rd exon of the microcalyx gene and essentially knocks out the entire locus from the fused message. And my project this summer was to analyze and compare carotenoids in each of these CRISPR lines, as well as in the RIN mutant and the wild-type tomato, at breaker plus 8 and breaker plus 15. And the breaker stage is when the fruit initiates ripening. And so 8 days after that is when the wild-type tomato typically becomes red and ripe. And so we took samples from that stage, as well as 7 days later at breaker plus 15. And so the results here came back from HPLC analysis of carotenoid levels. And you can see in the breaker plus 8 stage, the CRISPR line that targets the 3rd exon of the microcalyx locus has about half as much lycopene as the wild type. And lycopene is the red pigment. It's a carotenoid and it makes a tomato red. And so there's about half as much lycopene as the wild type in the 3rd exon CRISPR line. But in the CRISPR line targeting the EAR domain, a lot of these carotenoid levels are very similar to the RIN mutant. And so at breaker plus 15, the 3rd exon CRISPR line has just about the same amount of lycopene as the wild type, whereas the CRISPR line that targets the EAR domain remains, as you can see, it's yellow in this phenotype, and it contains similar carotenoid content to the RIN mutant. So these results suggest that the putative claim that the EAR domain is what represses ripening in the RIN mutant is actually false, and it's actually the entire microcalyx region of the fused message that is inhibiting ripening in this RIN mutant. I was surprised how much independence you have in a lab setting, and when we did the lectures about the PhD programs and grad school, which wasn't really related to the high schoolers, but I still learned a lot about what it takes to get a PhD. Sounded very intimidating, like doing your own research and finding new things out for yourself. And after that, I was like, I would never want to get a PhD. But I think this definitely at least made me realize that you can do your own research and come to your own conclusions.